Hey, my name's George, and welcome to this in-depth lesson on protein synthesis. What protein synthesis is a vital process that happens in every living cell, enabling our bodies to grow, repair, and function. Today, I'll take you step by step through how our cells convert the genetic information stored in DNA into proteins. The molecules that do the most of the work in our bodies will cover the two main stages of protein synthesis: transcription and translation. And by the end of this lesson, you'll have a deeper understanding of how these processes work together to sustain life. Let's start with an overview. Protein synthesis is the process by which cells build proteins, the building blocks of life. Proteins are made up of smaller units called amino acids, linked together in specific sequences. This sequence is determined by the genetic code in DNA. This process has two major steps: transcription and translation. Transcription happens in the nucleus, where DNA's instructions are copied into a temporary messenger molecule called mRNA. Then the next step, which is translation, this occurs in the ribosome, where the mRNA is read to assemble amino acids into a functional protein. Before we dive into the details, let's understand why DNA is important. DNA is like a library of instructions for building proteins, with each gene serving as a specific recipe. However, DNA itself stays protected in the nucleus and doesn't leave to interact with the rest of the cell. Instead, it sends out mRNA as a messenger, which carries the genetic code to the ribosomes. Now, let's look closely at transcription, the first stage. Transcription occurs in the nucleus, and here's how it works step by step. The first step is unwinding the DNA. The DNA molecule unwinds at the section where a specific gene is located. Only one strand of DNA, called the template strand, is used for transcription. The next step is RNA polymerase. An enzyme called RNA polymerase attaches to the DNA and begins reading the template strand. As it reads, it assembles a complementary strand of RNA called messenger RNA, or mRNA. For example, if the DNA has the sequence ATGC, the RNA polymerase pairs it with UACG in the mRNA, placing thymine with uracil. Now the third step, finalizing the mRNA. Once the gene has been transcribed, the mRNA strand attaches and exits the nucleus through a nuclear pore. The DNA rezips itself to maintain its integrity. The mRNA now carries the genetic message to the ribosome. Next comes translation, which happens in the ribosome, located in the cytoplasm. This is where the mRNA's message is used to assemble amino acids into a protein. Let's break this process into steps. The ribosome reads the mRNA in sequences of three bases, called codons. Each codon specifies one amino acid. For example, the codon AUG signals the start of protein synthesis and codes for the amino acid methionine. Next is transfer RNA. tRNA plays a critical role here. Each tRNA molecule carries a specific amino acid and has an anticodon that matches a codon on the mRNA. For example, if the mRNA codon is AUG, tRNA with the anticodon AUC will bring methionine to the ribosome. Next, building the chain. As the ribosome moves along the mRNA, tRNA molecules bring amino acids one by one. The ribosome links these amino acids together with peptide bonds, forming a long chain called a polypeptide. Next is folding into a protein. Once the entire mRNA is read, the polypeptide chain detaches and folds into a specific three-dimensional shape, becoming a functional protein. The shape of the protein is crucial because it determines its role, whether it's an enzyme, a structural component, or a signaling molecule. Protein synthesis is not just a fascinating process, it's essential for human life, or it's essential for any life. Proteins are involved in nearly every biological function. They make up the structure of your muscles and skin, carry oxygen in your blood, act as enzymes to speed up chemical reactions, and even help defend your body against infections. Without protein synthesis, our cells couldn't repair themselves, maintain homeostasis, or respond to changes in our environment. To summarize, protein synthesis is a two-step process. In transcription, DNA's instructions are copied into mRNA in the nucleus. In translation, the ribosome reads the mRNA, and tRNA brings amino acids to build a protein. This elegant and efficient system ensures that our cells produce the exact proteins they need to function. Thank you for watching this lesson on protein synthesis. If you found it helpful, feel free to share it. And don't forget to reach out if you have any questions or want to explore some topics in biology.